Artificial selection of animals and plants led to changes on particular genomic regions that affect traits of economic interest, as well as traits involved in adaptation to climatic and stress conditions, immune response, and disease resistance. Thus, selection signature regions are printed along the genome as a result of selection pressure. Detecting selection signatures is important for a better understanding of population history and genetic mechanisms affecting phenotypic differentiation in humans, livestock, and wild animals. Understanding how selection acts on livestock or plant populations may also benefit breeding programs in order to improve traits of economic interest in these animals and plant, such as chicken breeds which have been intensively selected for fast growth and muscle development. Many statistical approaches have been proposed to detect genetic signatures of selection. Several of them are based broadly on two concepts: the identification of allelic frequency differences between populations, and the detection of long haplotypes with or without differentiation of the ancestral allele. Direct observation of differences in allelic frequencies between populations is a rough way to identify loci showing differentiation. A more descriptive statistic and measure of genetic differentiation is fixation index, called abbreviated as FST. It is the most commonly used method since it is directly related to the variance in allele frequency between populations. This video will guide you through the steps for the identification of signatures of selection using FST and smoothed FST based on SNP allele frequencies. We will use the statistical programming language R. To calculate FST, we will need the allelic frequencies in each population. Depending on the format of the genotypes, different approaches can be taken to convert genotypes into frequencies. But this will be rather data problem specific. To illustrate, we will use three very distinct cattle breeds: Hanwoo, Angus, and Brahman, with 25 animals genotyped per breed on a single chromosome, nearly 8,000 SNP. The genotypes are in a plain text file, coded as 0, 1 and 2, 4, respectively, AA, AB, and BB alleles. The files are organized 1 SNP per line, and 1 animal per column. To read in the data in R, use the following command. As you can see, there are 7,763 SNP and 75 samples. The data is already organized so that the SNP are ordered based on their physical location on the chromosome, and there is no missing data. Ideally, no SNP that failed should be included in the analyses. For the samples, every 25 are animals from one of the breeds: Hanwoo from 1 to 25, Angus from 26 to 50 and Brahman from 51 to 75. Note that the genotypes used herein are for illustration purposes only. They are a small subset from the bovine 700 KSNP panel, and in high linkage disequilibrium, which is not desirable for these kinds of studies. The first 5 SNP samples look like 1. To calculate the frequencies in each population, first create a matrix with 3 columns to store frequencies of each SNP in each population, and compute allele frequencies as follow. Now, to calculate FST, you need to have average allele frequency across populations, deviation of each population from mean, and deviation squared divided by variance. Let's visualize the FST values for all 3 breeds. There is a lot of noise in the FST values we just calculated. A smoothing algorithm can help filter this out too. Identify more robust trends in the data. A useful R package for smoothing is Lockern, which can be used to calculate smoothed FST values for each population. The SNP data is already ordered based on chromosomal location. But if it was not, an additional parameter could be added too. Lockern is a vector of indexes for reordering the SNP. This could be based on the SNP map information. The N-out parameter defines the number of points to which the dataset will be smoothed to. In this case we used 77, which is approximately 1 point for every 100 SNP. The output from the smoothing process has a few items. The most important at this genome-wide plot of FST and smoothed FST, thick line. Notice how the smoothed FST highlights a region under potential selection around SNP 3500. It is less clear just from the FST due to the strong fluctuations from SNP to SNP.